Hello, everyone. I'm Shannon Wyman, Director of Program Development at Keystone Symposia, and it's my pleasure to welcome you to today's ePanel event. Today, we will feature the awardees of the Michelson Prizes Next Generation Grants, an international prize that supports early career researchers who are applying disruptive concepts and inventive processes to advance human immunology, vaccine discovery, and immunotherapy research. The prize is a collaboration between the Michelson Medical Research Foundation and the Human Immunome Project to award $150,000 research grants annually to support young investigators from a wide range of disciplines. Here to share a few remarks about the prize is Alya Michelson, co-chair of Michelson Philanthropies and Michelson Medical
When
Research
Gary and I
Foundation.
first dreamed up this prize, we were thinking about the young scientists out there, brilliant minds with big ideas who just needed someone to believe in them, someone to open a door. Together with the Human Immunome Project, we created the Michelson Prizes to do exactly that, to support fearless researchers who are ready to change the world but might not have the connections or resources to do so. This year, winners are incredible. Their work in immunology and vaccine research gives me so much hope, not just as someone who believes in science, but also as a mother and woman who cares deeply about the future we're all building. Immunology touches everything, from the way we fight disease to how we understand the human body, and I truly believe it holds the key to a healthier world. To all the recipients, I see your courage, curiosity, and commitment, and I am so proud to support you. Thank you to the Human Immunome Project, to our team, and to everyone who shares this mission. Congratulations to this year winners. I can't wait to see what you do next.
Great. Thank you, Alia. Next, we have Dr. Wayne Koff, founding partner of the Next Generation Grants, to provide a few comments about the goals of the prize.
Thank you, Shannon. It's wonderful to be here. And as Alya Michelson has just pointed out, when these prizes have begun 8 years ago, we had a discussion with the Michelsons about how they could really make an impact. And we honed in on the issue of the young investigator grants because at that time, the average age of the investigators who would receive an NIH grant was over 40. And of course, now in these days, when the research agenda at NIH is under siege, it's even more important that the action of the philanthropists can really make an impact. So this, as I mentioned, is the 8th year of the prizes. It was our most of the competitive a year. We had over 400 applications from either 69 or 70 countries. And the winners speak to the objective of the prize, and that is to be interdisciplinary, high risk, the potential for really high return across a range of the diseases. So I want to just go ahead and and offer the congratulations on behalf of the Human Immunome Project. The portal is now open again for the upcoming year of the Michelson Prizes. So if you're a young investigator out there who's got an interesting idea in immunology, in vaccines, and immunotherapy that crosses a range of diseases, we want to hear from you. So again, I thank the Michelsons for the visionary philanthropy, and I look forward to seeing the videos of the winners. Thank you, Shannon.
Great. Thank you, Dr. Koff. And now it's my pleasure to introduce this year's awardees and their groundbreaking work to advance our understanding of human immunology and applications towards cancer vaccines and immunotherapies. After I introduce each awardee, we will show a 10 to 15-minute presentation about their work. Once all of our awardees have presented, we will have a collective group Q&A. You can submit your questions at any time into the Q&A box and vote on others' questions you would also like to hear answered. Please note if your question is directed for a specific speaker in the text of your question. And now I'd like to welcome our first awardee, Dr. Caleb Lareau.
Thanks, Shannon. It's really an honor to be recognized with this award. It's really a phenomenal honor and very much looking forward to the feedback and questions from those in the audience. So thank you all for joining online.
Thanks, Caleb. Caleb is an assistant member of the Computational and Systems Biology program at Memorial Sloan Kettering Cancer Center. Caleb studies the human virome, documenting millions of viruses that live within us and the impacts they have on our health, disease susceptibility, and outcomes. In particular, he aims to reveal how these endemic viruses, which seemingly cause no ill effects, actually shape systemic immune responses with the potential to trigger autoimmunity or influence our ability to fight cancer. To do so, he will develop novel data science tools to analyze biobank information to map how the human virome shapes complex diseases. His proposal is titled How the Human Virome Influences Health.
Hello, my name is Caleb Lareau, and I'm an assistant member in the Computational and Systems Biology program at Memorial Sloan Kettering Cancer Center. I'm really honored to be a recipient of the 2024 Next Generation Grant from the Michelson Research Foundation and the Human Immunome Project, and I sincerely want to thank these organizations for their kind consideration of me and the award for my research group. Today, I want to tell you a little bit about the work that we've been pursuing and specifically those projects that are going to be enabled by this award. What my lab studies more than anything else is really this fascinating accumulation of DNA sequencing data on the planet. And so here you can see some schematics of the amount of DNA sequencing that is present in the largest public repository, the Sequence Read Archive, the accumulation of millions of single cells that have been profiled, as well as hundreds of thousands of whole genomes that have been sequenced in the biobank. And roughly speaking, this synthesis of data is accumulating at the zettaBase scale, such that in 2030, we will have sequenced somewhere in the order of 10 to 21 bases, or zettaBase. And for perspective, this is consistent with the amount of grains of sand that are on the planet would be commensurate with the amount of DNA bases that we've measured. So it's really this massive volume of data that is the primary substrate of our research program. And so what can you do with just a sheer volume of DNA sequencing data like this? One of the things that's really exciting is the capacity to discover novel and known viruses that potentially are impacting human health. And so there's been a few transformative papers that have come out in the past few years doing viral discovery on this massive amount of DNA sequencing data. One of these resources is from a colleague of mine, and the tool is called Ceratus. And so what Ceratus does is it looked at all public sequencing data with the goal of trying to identify novel viral species using a heuristic that basically said, okay, we can take a set of viruses that we know, somewhere about 10,000 of these that exist, and then we're going to go search for motifs that are consistent with something that we have prior knowledge of. And after a 10-day computational search, they were able to identify over 130,000 novel viral species. And again, the way that this was achieved is by looking for conserved motifs within the RDRP domain, a sort of critical protein required for our viral RNAs to function. And so by doing this, this paper really looked for sort of planetary scale evaluation of potential novel viral species. And so I remember reading this paper in my postdoc and being really taken by the idea that we could just go back through existing data and find novel viruses or novel viral associations. And that was really the genesis for my work back at Stanford when I was a postdoctoral associate in Ansu Sapathy's lab. So as an immunologist, I was specifically interested in potential viruses that were associated with T cells, sort of this classically functional cell type that is showing immense promise also for potential new immunotherapies. What we discovered was that HHV6, a endemic human herpes virus, was routinely identified in RNA and DNA sequencing data sets that were derived from T cells. And we're able to figure this out just from a few-minute query using the Ceratus tool. So whenever we went in and looked at these old sequencing data sets that had HHV6 annotations, what we discovered was that the simple act of culturing memory CD4 T cells, just with activating CD3, CD28, and in culture with IL-2, routinely over this multi-week culture duration, you would see HHV6 becoming reactivated. And so here I'm showing an experiment that was done by colleagues at Scripps. They had no idea that there was any virus in their T cells, But what happened was in one of the four donors of those cell culture lots, about 4% of all RNA molecules by the end of the 2-week culture was actually derived from the virus as opposed to the human, essentially indicating that the simple act of culturing these T cells was reactivating this herpes virus at extremely high levels. And this wasn't something that was strictly happening at Scripps. The same thing also happened in Heidelberg with a similar experimental design. Again, just the simple act of culturing T cells led to large-scale human herpes 6 reactivation. And so this is potentially quite relevant because we wanted to know, is this also then happening in therapeutic T cell cultures? And to make a long story short, the answer is yes. So we did some additional work profiling research-grade CAR T cell therapies longitudinally. And indeed, what we could see was that somewhere between 10 days and 2 weeks of standard kind of CAR T manufacturing and culturing, there would be the reactivation of this virus. And this wasn't just happening in a dish. This is something that we could actually see in products that have been going into patients, including data from a phase 1 clinical trial where we could detect this reactivated herpes virus in an infusion product, and then ultimately detecting additional HHV6 positive, CAR positive cells in an individual, ultimately showing that we could potentially identify these reactivated herpes virus cells coming from the cell therapy manufacturing process itself. But what I want to emphasize from this work is not so much the T cell phenotype or the CAR phenotype, but really it's just this way of thinking that as you go and manufacture cell therapies, there's all kinds of potential for things to go wrong. And in this case, we happen to find HHV6 in T cells, but really there's going to be a variety of viral nucleic acids that are going to show up that could potentially impact the product efficacy or potentially impact patient safety. And so roughly speaking, this has been what I've been thinking about for the last several years. And then as I've been able to transition to start my own group, it's really been motivated by the idea that we can find these novel and exciting associations between the host and virus just by querying large data sets. And so where my work has taken us now is both trying to look at known viruses that we have a good sense as to what they are, things like HHV6, where we could potentially associate these viruses to novel human phenotypes, but also potentially discover novel viruses, things that we don't have a good idea that as to what they are, what they do, but potentially these are actually driving some degree of human phenotypes. And so really what my lab is focusing on right now is developing computational molecular tools to be able to study these known and novel viruses. And so where we're focusing now, enabled by this Michelson Award, is focusing specifically on what's called the human blood virome. And so this has been a concept that's been around for a few years now. So there was a really formative paper that came out 7, 8 years ago now, basically showing that you could take whole genome sequencing data from large cohorts and using all the unmapped reads could identify all sorts of viral species, again, the so-called blood virome. So we certainly see tons of human herpes viruses. We see a variety of phages, as well as a variety of other viruses that are just present in humans' blood, readily detected by standard DNA sequencing tools. So the mental model here is that we as humans are kind of constantly being bombarded by these endemic viruses that are present in our environment. Most of the time, our immune system is able to control that, but there are instances where these viruses can then convert to latency, and then ultimately they're sticking around, potentially causing havoc in our immune system in ways that we don't fully understand yet. And so what we've been doing are taking these very large cohorts that have now been online for a couple of years now, resources like the UK Biobank and the All of Us cohort from the United States, where they've done whole genome sequencing on hundreds of thousands of individuals. And what we do is take these blood-derived whole genome sequencing profiles and we take all the junk, all the discarded reads that don't map to the human reference genome, and we ask the question, do any of these things look like viruses? And whenever we do this computation, we can then identify individuals that have varying levels of viral DNA in their blood and use this as a novel molecular biomarker for doing both genome-wide association studies as well as phenome-wide association studies. The idea being that potentially different genetic variants between all of us in the population may control how we are all responding to the same sorts of viral infections, as well as those of us that are retaining high levels of viral DNA in our blood. Potentially, this is a biomarker of diseases of some sort. So just to give a flavor for what this data is starting to look like, we've done this now for EBV, which is another herpes virus that's also endemic. By quantifying this in the UK Biobank, we're able to say how much EBV DNA is in everyone's blood, and then we do a genome-wide association study. So here each dot is a different genetic variant, and I've annotated the proximity to genes that are statistically significant, with the goal of hopefully understanding what are the genetic determinants of how we're processing these EBV infections. And what we found are about 25 conditionally independent loci that attain genome-wide significance. These include antigen processing factors, things like the HLA region, as well as transcription factors that certainly play a role either in the primary processing of the virus or its control in adaptive and innate immunity, as well as a variety of signaling molecules, basically suggesting to us that there is indeed genetic determinants of how we are responding to the same infectious agent. But where we're going to go with this grant is a little less focusing on herpes viruses and to something that is a new species that I've been learning a lot about recently, and it's called anelloviruses. So these anelloviruses are actually the most abundant part of your blood virome, and they're also the sort of most diverse viral species that we know to infect humans. Here you can see a schematic of these anelloviruses and their diversity relative to other viruses you may be more familiar with, such as APV and AAVs. And you can appreciate that the anelloviruses are just so much more diverse. There's three major clades, alpha, beta, gamma. But what I want to really emphasize is that I think there's kind of emerging evidence. Here's a table from Biobank Japan showing that anellovirus viremia, or the ability to detect anellovirus in your blood, is massively associated with a variety of autoimmune diseases, basically suggesting that this viral species that we've only known about for about 20 years could be a potential risk factor or biomarker for autoimmunity in ways that we're not yet fully able to appreciate. And so beyond just finding these viral sequences in existing data sets, our lab is also taking a deliberate interest in going from what we call computer viruses to sort of novel molecular agents actually that we can manipulate in the lab. And so one of the tools that we've been working with is a new resource called PerfSeq. This is the first research paper from my group, where ultimately we're able to identify cells that are harboring specific infection via hybridizing to arbitrary RNA species. And this is really useful because oftentimes for these newer viral species, we don't have good antibodies to be able to isolate and study these viruses in any appreciable way. And so roughly speaking, PerfSeq just allows you to hybridize fluorescent probes to whatever RNA species you want. And then after doing flow cytometry, you can study those cells that have been infected with single cell genomics. And just to come full circle with the original interest in HHV6, what we're able to now show is that we can hybridize two cells that have been variably infected with HHV6. The signal is super clean. We're able to identify cells that have different abundances of HHV6 RNA corresponding with different stages of the viral life cycle. And so just to summarize this research vision and where we're going to go with this award, I just want to emphasize that all of your sequencing libraries have some degree of viral DNA and RNA in there, whether you know it or not. And this is just due to the sheer volume of viruses that are present in our environment. One specific example that we've already found is that CAR T cells themselves can harbor HHV6, and this may or may not have potential clinical implications as a function of if this virus is reactivating in these cell therapy cultures. Ultimately, where we're going to continue to push this is in retrospective data analysis to find new associations between non and novel viruses. But then ultimately, this prize is going to allow us to do both these large-scale data analyses as well as novel molecular tool development to ultimately, hopefully, study these in greater function. The goal ultimately being to find new biomarkers and new biology that really refine our understanding of host-viral interactions. With that, I want to thank everyone who joined my group. I was able to start in New York about a year and a half ago. I also want to give a big shout out to Ansu Satpathy, who's been my mentor during my postdoc period and a previous recipient of the Michelson Prize, as well as my other funding sources at MSK. And with that, I'm happy to take any questions at the Q&A that'll happen a little later.
Great, thank you, Caleb. Our next awardee is Dr. Yzhong Liu. Thanks for joining us, Dr. Liu.
Hi, everyone. Hi, my name is Yzhong, and I would like to thank the organization for the recognition of my work and the opportunity for me to share with you a little bit of my work today.
Great. Yzhong is an assistant professor in the Department of Chemistry at Scripps Research Institute. Yzhong is working to improve the efficacy of cancer vaccines. Currently, cancer vaccines are limited by the inability of tumor antigens and current adjuvants to stimulate adaptive immunity. Yzhong aims to design better adjuvants by using synthetic biology to study how adjuvant structures correlate with their immunostimulatory capacity. By identifying structural features that stimulate adaptive immune responses, she aims to guide the informed design of more potent and safer adjuvant candidates for cancer vaccine applications. Her proposal is titled Improved Saponin-Based Adjuvants for Cancer Vaccines.
Hello, everyone, and thank you for being here. My name is Yzhong Liu, and I am an assistant professor in the Department of Chemistry at Scripps Research. I'm incredibly honored to be a recipient of the 2024 Michelson Prize Next Generation Grants, and this award allows my lab to pursue bold and foundational work at the intersection of synthetic biology and immunotherapy. Today, it's my great pleasure to share with you our approach to biosynthesize safer and more potent saponin-based adjuvants for cancer vaccines. Let me start by explaining the problem. Modern cancer vaccines are improving, but cancer antigens alone are poor at stimulating adaptive immunity. And that's exactly where adjuvants come in. So adjuvants are the molecules that can boost your immune response, thereby reducing the amount of antigens necessary, and can also enable long-term memory, all of which are especially important in populations with weaker immunity, such as the elderly or immunocompromised. So the figure on the left shows the effect of adding an adjuvant to a vaccine compared to in the absence of. So here you can see that the adjuvant triggers a cascade of innate immune signals, recruiting more antigen-presenting cells, stimulating cytokine release, and enhancing antigen uptake. And these mature antigen-presenting cells prime both the CD4 helper T cells and the CD8 cytotoxic T cells, leading to a much more robust and balanced immune response. And shown here, the outcome is not just more antibodies, but also the activation of cytotoxic T cells, formation of plasma cells, and expansion of memory B cells. All of these are absolutely critical for effective and long-lasting protection, especially in the context of cancer immunotherapy. As a result, the global vaccine adjuvant market size was about $1 billion in 2023, and is expected to register a rapid growth to reach $2.7 billion in 2033. Despite their importance, the number of adjuvants that have been clinically approved is actually surprisingly small. Alum, introduced in the 1920s, is still the most widely used adjuvant, but it's weak at inducing cellular immunity, and it's not until recent decades that MF59 and AS03, both oil in water emulsions, were approved for flu vaccines. AS01, which combines QS21 and MPLA, is used in the GSK shingles and malaria vaccines, and CpG 1018, a TLR9 agonist, was approved for hepatitis B vaccines. And the limited amount of vaccines adjuvants that are available to us is actually due to the lack of a systematic platform to explore and optimize adjuvant structures. And that means that each candidate must be discovered, isolated, characterized, and tested individually. And this is exactly the challenge we're trying to solve here with a biosynthetic strategy that enables modular production for structure variation to really understand the structure-activity relationship for their immunological outcomes. The molecule that we became very interested in during my postdoctoral research is a saponin called QS21, of which the structure is shown here. You can see that it's made of a functionalized triterpenoid core, shown in black in the middle, before it's functionalized by a branched trisaccharide, so 3 sugars on the left, shown in blue, and a linear tetrasaccharide, 4 sugars on the right, shown in green, and before it's further decorated by 2 repeating acyl units made of 9 carbons, and the last sugar addition. So you can see a pretty sweet molecule overall. And it's an FDA-approved ingredient in 3 vaccines already. And in addition to meeting its increasing demand for the existing vaccines, the gap between demand and supply is especially pressing given the growing interest in personalized cancer vaccines
and the pandemic preparedness to be able to supply millions of doses on a single day. And to be able to scale these efforts, we need a robust synthetic platform towards the development of safer, tunable, and synthetically accessible vaccine adjuvants. While QS21 is great, harvesting sufficient amount of it turns out to be quite difficult. And traditionally, it's extracted and purified from Quercus saponaria, which is this exotic tree that only makes QS21 in specific areas in Chile. And it's extracting one molecule out of an entire plant, as you can imagine, is rather laborious, low yielding, and not to mention, it's not very sustainable. And the Herculean effort of the total synthesis of QS21 has been achieved by Professor David Jin's group in the early 2000s. And starting from this trisaccharide intermediate, which is more abundant in the tree, which means you still have to extract from the tree, it still takes more than 76 synthetic steps with an overall negligible yield to get to QS21. And because of the difficulty in sourcing, QS21 is currently priced at more than $1,000 per milligram. With all the genetic modification tools that are available to us and the advancements in sequencing technologies, we can really start to think about how to make microorganisms to do things that they wouldn't naturally do. So in this case, you can imagine to copy the biosynthetic gene cluster that's responsible for making QS21 from the plant, and then paste that into the genome of a genetically tractable microorganism such as Saccharomyces cerevisiae, baker's yeast, and with a production scheme that's pretty similar to how you would make beer and bread at home by feeding in simple sugars, wait for a couple of days. You will be able to access this exact natural product in a couple of days rather than decades. You can also imagine to swap in and out some of the pathway enzymes to also build a library of unnatural structure derivatives to establish structure-activity relationship to fully understand the mechanism. And last but not least, the production is now easily scalable. To show you what this plant biosynthetic pathway of QS21 looks like and how we installed it into a microorganism is that we hijacked the mevalonate pathway, which is native to yeast, that converts glucose to acetyl-CoA, and via an upregulated isoprenoid pathway, we were able to access high levels of 2,3-oxidosqualene, of which the structure is shown here. And that followed by a cyclase that folds this long-chain hydrocarbon into a pentacyclic terpene backbone beta-amarin, which is then oxidized by 3 different P450s at 3 different carbon positions to install a carboxylic acid, an aldehyde, and a hydroxy group. Now we have quillic acid, which is the necessary terpenoid backbone for glycosylation. And the later synthetic steps turn out to actually follow a highly orchestrated manner to install the 3 sugars on the left and the 4 sugars on the right before the 2 azo units can be added sequentially by 2 different azo transferases. And with the last sugar addition, we finish the main biosynthetic pathway. After expressing all of these enzymes into one single yeast strain, we were very happy to see that we were able to produce QS21 that co-eludes with the commercial standard. And the synthesis was further confirmed by the identical isotopic fingerprint pattern that is shown on the left. To summarize this part of the work, we were able to achieve the complete biosynthesis of QS21 in yeast by reconstituting 38 heterologous genes from various enzymatic families while overexpressing 8 yeast genes to maintain its native metabolism. And many of these genes actually come from 6 different species, varying from trees to fungi and bacteria, to kind of harness nature's best activity into one single organism. Many of you might have noticed that there's a small peak in addition to the major QS21 peak that was identified in the LC-MS spectrum. And it took many iterations of in vitro glycosylation assays to identify that this is actually a terminally xylosylated version of QS21 rather than the original arabinofuranosylated QS21. And this tells us that these glycosyl transferases may not be as stringent as we traditionally think that they are. Indeed, as this blog on Science pointed out, that there is no particular reason why QS21 itself should be the best adjuvant out there, and this will be just a way to study its structure-activity relationship to see if there's anything safer and better. With this microbial biosynthetic platform that we have built, we can now unlock the ability to systematically tweak and test these saponin structures. And by using native and engineered glycosytransferases, we can reconstruct saponin frameworks such as QS21 with a structure-guided engineering approach to enable the biosynthesis of unnatural glycan sequences that are inaccessible via chemical synthesis or natural extraction before. And this approach also allows us to systematically explore glycan structure-activity relationships to uncover how individual sugar moieties can modulate immunostimulatory activity, toxicity, and pharmacokinetics. And in the long run, we're hoping to build a mechanistically informed platform to guide glycodiversification of saponins towards more potent and safer adjuvants for cancer immunotherapy that can be made available and affordable to everyone. With that, I would like to thank the incredible team that made the QS21 work possible, my postdoctoral advisor, Professor Jay Kiesling, our collaborators, and the entire QS21 team. I'm also especially proud of my current students and postdocs for doing all the groundbreaking work, both in the literal and metaphorical sense, starting a new lab. I would also like to convey my special thanks to my collaborator on this project, Professor Darrell Irvine, for his insights on cancer immunotherapy, and to Professor Peter Schultz for providing lab space and a home for my new group in this beautiful new building. And finally, I'm truly grateful to the Michelson Foundation for supporting early stage ideas like this one, and thank you all for your attention.
Thank you, Yzhong. And now I'd like to welcome our third winner today, Dr. Omar Abudayyeh. Omar, welcome.
Yeah, thanks so much for having me, and
You're
thank
muted.
you. Am I muted?
Nope, you're good.
Hello?
Sorry.
Oh, okay. Yeah, thanks so much for having me today. Yeah, super grateful to the Michelson Foundation for supporting this type of research, and thank you to the Keystone Group for putting this all together today.
Great. Omar is an assistant professor at Harvard Medical School and faculty member in the Department of Stem Cell and Regenerative Biology at Harvard University. He is also an investigator at Brigham and Women's Hospital and Mass General Brigham's Gene and Cell Therapy Institute. Omar is working on a revolutionary new approach to cancer immunotherapy by using mRNA to deliver the immunotherapy molecule within tumor cells. Current immunotherapies, such as antibody-drug conjugates, are ineffective against intracellular targets because they're too large to get inside the cell. Omar's new approach will provide a means to manufacture such cytotoxic factors and other immunotherapy agents precisely within the tumor cells themselves based on their transcriptional state. By leveraging the potential of mRNA therapies, Omar aims to enhance the efficacy of cancer immunotherapies while also reducing the risk of side effects. His proposal is titled Development of Programmable RNA Sensors for Next Generation Cancer Immunotherapies.
Hello, my name is Omar Abudayyeh, and I'm an assistant professor at Harvard Medical School. And today I'll be talking about some of the work we're doing to program and manipulate cell states based on transcriptomic signatures and specific RNA markers. So just to dive in, a big question our lab likes to tackle is biology programmable, and can we build the right tool set to be able to manipulate it in precise ways? And I'm sure we're all familiar with these very complex diagrams of cell circuits and cell states, and it makes it seem like biology can be very intractable and complex. But we think through very different types of programmable technologies that we can break down this complexity, and we do this in sort of three buckets. We try to engineer and program proteins and molecules historically, right? That's looked like through CRISPR and RNA-guided genomic engineering technologies. We're really interested also in mapping cell states and developing tools to convert between specific cell states and across the whole landscape of different types of cells that we observe. And lastly, building it up into sort of mechanisms of manipulating whole organisms and specifically studying aging and rejuvenation. And so in actual concrete terms, we do a lot with genome editing and protein engineering approaches, different ways of cell reprogramming, whether it's with transcription factor screens or computational modeling of cell states and ways to manipulate them. And lastly, through very precise molecular characterization of aging and trying to come up with ways to manipulate those processes. And so for this talk, I'll focus specifically on ways we can program cell states. Historically, folks have had to do very sort of brute force approaches to engineer cell states, whether that's screening transcription factors or small molecules and combinations of medias and factors to manipulate differentiation or transdifferentiation. But we've long looked for ways to sort of report and manipulate cell states that could look more like CRISPR technologies, where you have an RNA-guided approach that's very programmable, where you don't have to think about what you're doing. You just program against the complementary sequence and you're good to go. And because single cell sequencing has become so mature and we have very precise ways to define cell states and cell types with RNA markers, we've longed for a way to do this based on RNA markers of cell states, essentially using some sort of sensor that can take one or more markers and output any sort of output you want, whether that's, just a reporter molecule like a GFP, or it could be, transcription factors or cytotoxic payloads to kill cells, etc. One of the ways we've tried to do this is by leveraging ADAR enzymes. We've used ADAR enzymes for a long time for RNA editing and therapeutic approaches, but we thought that maybe we could actually leverage them for this purpose of sensing RNA states and then doing something. And ADARs are a diverse family of enzymes. There's a few different classes, ADAR1, ADAR2, ADAR3, but typically they look like some sort of deaminase domain fused to an RNA binding domain. And essentially they help catalyze this adenosine to inosine reaction through a specific deamination of the base. And like I said before, this has been very popular for therapeutic RNA editing because inosines are read out as guanines by the ribosome, allowing for specific protein level changes. And we actually wondered whether this paradigm could be inverted to allow for RNA sensing. And the idea here is we can basically take a payload, this could be a therapeutic payload, a GFP, or whatever you want to gate based on a cell state, and we put a stop codon in front of it. And the stop codon blocks translation of this protein payload. And we designed the stop codon context, basically the surrounding nucleotides, to be complementary to a specific target transcript. So let's say you want to target regulatory T cells. A good marker for regulatory T cells is FOXP3, which is an intracellular marker that's overexpressed. And we can basically make this complementary to that gene. The gene's expressed, then we'll form an RNA duplex in the cell. We design this duplex in such a way that it's ideal for recruitment of an ADAR, and that can be an ADAR we supply, or it could be endogenous ADARs. And that allows for editing of the stop codon, which causes read-through, and thus then translation of the protein. And so in a very simple way, we've connected detection of an RNA marker to output of a generic protein, which is a very powerful concept that allows for programmability and RNA-guided cell state targeting. We call this radars. And so just to give some examples of, radar approaches, here we're just detecting an overexpressed target transcript, I think IL-6 in this case, and we output a GFP protein. And you can see when we overexpress ADAR, we get very nice output of the GFP across the cells. And if we don't overexpress ADAR, we can still get low levels of fluorescence due to the endogenous ADAR, allowing for editing. And if we don't have the target, we don't see editing. We also can enable these sensors to be quantitative. And so this is showing the output of the radars on the y-axis and the target that we can modulate its expression level on the x-axis, and the qPCR is its expression level. And you can see that we can get a quantitative response. Now, this is all with overexpressed transcripts, so we wondered whether we could also sense endogenous transcripts, which is ultimately what we want. And so we came up with a way to modulate endogenous transcripts using siRNA, and then could read out detection of the transcript with radars. And so the idea here is we took 6 different genes that are expressed from very low levels, TPMs of 13, to very high levels of TPMs of almost 8,000. And we do a dose titration of the siRNA on the x-axis to allow for modulation of the gene level. And we do a targeting sensor, a non-targeting sensor, and then just the orange line is the actual expression of the gene due to the siRNA knockdown. And so you can see across these genes, we can get very nice relationships between the targeting sensor and the qPCR level of the gene. Starts to break down maybe a little bit as you get to the KRAS because the expression level is very low. And the non-targeting sensor does not have a response. And so this shows that we can actually target a wide range of expression levels of endogenous transcripts inside the cell. Another common thing that we would want to do is a lot of cell states are actually marked by 2 markers, not 1 marker. And so if we want a AND gate, essentially, we can actually string 2 radar sensors together such that both stop codons have to be edited. And in this case, we only see output of the radars, which in this case is a luciferase activation when you have both targets expressed and very minimal background otherwise. So lastly, we wanted to do an in vivo demonstration to show that this could be used in the liver. And so we basically took a deep red luciferase that allows for in vivo luminescence imaging and gated it based on the presence of human SERPINA1. And that allows us to then use humanized SERPINA1 locus mouse for detection and compare it to a wild-type mouse that does not have the human SERPINA1 transcript. And thus we can inject these mRNAs. We actually make these as mRNAs, so we do in vitro transcription using T7 polymerase. Then we can package this mRNA with lipid nanoparticles and see if we get luciferase activation after 18 hours post-injection. We thus compared the 2 SERPINA1 sensors to a non-targeting sensor and a constitutive luciferase that has no sensor. And what you can see with the constitutive case, we see always turning on of luciferase. With the non-targeting sensor, we don't see turning on of luciferase. And then with the 2 human SERPINA1 sensors, we see turning on of the sensor inside the humanized mouse. And it corresponds to about 2 to 3-fold activation of luciferase. Now, I think in the future, we really would want this to be much higher. I should also say we're not overexpressing ADAR here. We're using endogenous ADAR, showing that this can really be used with endogenous levels. But with that said, we really need to improve the sensor. We need to improve how we deliver these things, and actually, we believe we can get very nice levels in vivo. I should also say Josh Huang from Duke had a nice paper where he showed with AAV they can get very nice brain targeting with these sensors as well. And so there's a lot of evidence that in vivo we can get these systems to work. With that said, one application area that's actually funded by this award is using this to basically do programmable cancer therapies with mRNA. And the idea here is there's been interest in using mRNA to essentially hone to tumors inside the body and heat them up in some way, whether it's expressing cytokines to sort of locally inflame the tumor microenvironment and recruit immune cells, or even cytotoxic payloads that can kill some fraction of the tumor cells. It causes release of all sorts of molecules that can inflame and recruit immune cells and lead to essentially immune clearance of the tumor. And so, one of the issues with this mRNA approach is it's very sensitive to off targets. You can't systemically deliver these things because they'll express in other tissues and you'll cause inflammation. And so typically you have to locally inject these mRNAs directly in the tumor. And so we wondered whether with radars we could actually allow for systemic injection, which would then allow only turning on of these sensors inside the tumor, thus giving us a nice therapeutic window for this approach. And the advantage here is, most tumors are not really accessible for local injection. Local injection doesn't also address, metastatic sites and areas you don't know where the tumor is. And so this could be a very powerful way of targeting tumors. And if we can actually target, markers that are somewhat conserved across different tumor states, we can actually develop semi-universal approaches that target all sorts of different tumors. And so just to show an early example of some of this work that's unpublished, we've developed sensors against liver cancer specific markers, specific to HCC. So HEPA16 is an example of a HCC cell line, and 3T3s are a control fibroblast line. And we can just show, using a cytotoxic payload, It's actually a combination of a cytotoxic payload and other cytokines. We can find sensors that actually can kill the liver cancer cells specifically and not affect the fibroblasts due to the radar sensor. And we can actually inject these in a liver cancer mouse model and find that we can get, with one of our sensor designs, increased sort of tumor reduction due to the combination of cytokines and cytotoxic payloads we use with the radar sensor. And this is very preliminary data, but this shows the vision we have to basically bring tumor-specific mRNA therapies forward and see if we can really target liver cancer and perhaps other tumors as well. So with that said, we're very excited about continuing to improve the designs and engineering of the sensors to get much better activation. There's a lot of engineering we do originally in the paper that you can see how we have different levers to improve these things, and then really applying this to cell state specific therapeutics in cancer, aging, autoimmune disease, and lots of applications beyond. For example, for cell specific lineage tracing and cell tracking. So I'm very excited to distribute this to the community, to collaborate on this approach, and make new types of therapies. So with that said, thank you to the many lab members that drove this work, including Kai from our lab, as well as folks from Fei Chen's lab, which is who we collaborated with on this technology, and of course all the folks in our lab and our many funders as well. So thanks so much for listening, and always feel free to reach out if you're interested in the technology or have any questions.
Thanks, Omar, and thanks for you all to join us today for your inspiring work and sharing that with the audience. I really look forward to the discussion with you all now. Reminder to our audience, please submit your questions into the Q&A tab and vote on others' questions you would like to hear answered. I'll start with some for our panelists, but first I'll start with a few questions that were submitted regarding the grants themselves, and then move on to the questions for panelist discussion. So one of our first questions about the grants is if you can apply for the Next Generation grant with just a master's degree, and the answer is yes. So if you have a master's degree but not a PhD or an MD, please do apply. And again, the portal is now open for this year's awardees. Second, do the research grants cover other areas of vaccine development beyond cancer vaccines? And again, the answer is yes. The Michelson Prizes are interested in receiving applications that address a wide array of diseases, areas related to immunology, including all types of vaccines and other immunology questions. So very wide array of research that they're excited to fund. Another question we have is, do you consider international researchers who are not UK or US citizens in your research grant award? And again, the answer is yes. The Next Generation grants are an international prize with no geographic eligibility criteria, which distinguishes it from a lot of other sources of funding. So they really do encourage researchers from around the world to apply. Great. Now we'll go to the discussion with our panelists. I'll start with a question for everybody. And the question is, how will the Next Generation grant help you accomplish your research goals? And why is the project that they funded unlikely to be funded by other more traditional sources? So maybe we'll start with Caleb.
Yeah, thanks for the questions, Shannon. Kind of what I was indicating from my talk is that I really have no idea what viral species or what disease I'm studying. I kind of do the everything, what's the movie called again? Everything Everywhere All at Once approach to this. And so it's really hard to convince a more traditional funding agency that not knowing the disease or not knowing the virus that you're going to characterize by the end of the project is typically a step too far away from sort of alignment with typical funding visions. And I really am grateful for the Michelson Foundation for recognizing the sort of transformative potential of this work, despite me not being able to put a lot of these nouns together that you would normally have to for a traditional approach. And so I think it's really that way of thinking that is pretty novel to the way that we approach viruses, and hopefully this is going to allow for these big insights.
Great. And Yzhong?
Yeah, so as you can probably tell from the presentation that was just shared, our work really lies at the intersection of synthetic biology, immunology, and glycoscience. And this type of interdisciplinary work is kind of, unfortunately, where a lot of these traditional funding mechanisms can fall short. A lot of these agencies will prioritize, more well-established systems or will require a lot of extensive preliminary data. But this is exactly where the Michelson grant actually comes in, and it can help us, de-risk these kind of bold and novel ideas, and then generate sufficient amount of data to hopefully pursue larger scale translational efforts in partnership with, industry as well as federal agencies. So yeah.
Great, thank you. And Omar.
Yeah, I think a lot of our lab's work focuses on technology and platform development, and I think with traditional funding agencies, what we found is you basically have to have the entire technology developed and completely worked out, and all the kinks ironed out before you can really get that funding. And I think Michelson is really helpful because it allows us, you essentially do that kind of work now, where we can actually, throw the kitchen sink at the wall and figure out, all the levers we can pull to get this technology working. And kind of similar to what Caleb was saying, where, if you propose this, it would just look like you're doing all sorts of different things, and people, won't be convinced that necessarily all those things will work. Although in practice, I think, as long as we keep iterating, we can see improvements in these types of technologies. So really grateful that the Michelson can see the promise of sort of an idea and a vision, and help us try to get to that point.
Great. Yeah, I'm sure the technology development takes quite a lot of troubleshooting to get to a good place. So great. So I'll ask each of you a question. Feel free to build on each other's answers and have a discussion about it. But I'll start with Caleb. You talked about the reactivation of the viruses in T cells. Are there other tissues that you think you're specifically likely to find these viruses? And depending on where they're found, does that have a likely impact on how they may impact health, like whether they're found in the gut, brain, the liver, the T cells, as you mentioned?
Yeah, it's a great question. So again, kind of our approach is really predicated on existing data, and so that's really been the strength of us focusing on the blood, just because there's petabytes of people's blood sequence, and so that really elicits a much broader substrate to find these entities. But if I were to have it my way and I could generate all the data I wanted, certainly the gut and the colon, the entire GI tract really is probably where there's the most consequential infection sort of dynamics occurring. Much like what we've discovered with the human microbiome, I think the human virome is probably highly dynamic there. So, the sort of two-sided answer directly is, I think the colon and the GI tract is probably where we would love to generate more data, and that'll happen at some point in the lab. But where we're going to go looking just purely based on convenience and really the fact that it's a pretty kind of new way of thinking, we think we're going to find plenty of things in immune cells still, and that's where we're going to go first.
And if you find it in the blood, is there a way to tell where it may have come from initially? Or no, you're going to cross compare what you find in the blood to other tissues?
Yeah. So there's, for these anelloviruses, for example, that are probably, again, they're the most abundant viral species in our bodies that are infecting eukaryotic cells, and they've been scarcely characterized. I mean, not so many PubMed entries about these. What we know about their tropism has been kind of from doing flow cytometry for well-defined immune subsets, and then doing a variety of PCR assays to try to quantify their abundances in these different subtypes. What we're seeing is that immune cells in different tissues have different compositions of anellos, different abundances. So I think there's already quite a bit of evidence for that. So that's certainly one approach that we'll consider. The other would be something kind of more modern, like a single cell sequencing approach, where you're trying to co-detect the viruses alongside with lineage-specific markers. So you could maybe both say, like, what is the cell that harbors this virus, as well as how is the cell responding to a viral infection being in there, but somehow maintaining latency. So we're certainly very keen on these sequencing-based approaches to discover this, and probably where we'll be spend time thinking about it.
Great. And Yzhong, you're looking at adjuvants to use for cancer vaccines specifically. What's the difference between adjuvants used for cancer vaccines versus infectious disease vaccines, and why it seems like maybe cancer vaccine adjuvants are more difficult to find or come by? Can you talk about that a bit?
Yeah, absolutely. It sounds like vaccines for adjuvants for vaccines should be all pretty similar, but for different vaccines, they actually require different things. So, for example, for infectious disease vaccines, they oftentimes, target pathogens that your immune system can tell that is clearly foreign. So, relatively mild adjuvants can already stimulate antibody responses and your T cell activation. That can be sufficient. But unfortunately for cancer vaccines, it's particularly challenging because the tumors that we're dealing with here oftentimes originate from your own tissues. And sometimes they can even develop mechanisms to suppress these immune responses that your body can generate, because it's your own. So these adjuvants for cancer vaccines will have to stimulate the immune system, not only to just elevate your response, but also to break the immune tolerance and overcome the immunosuppressive tumor microenvironment. And this will require much stronger adjuvants that are capable of, promoting cross-presentation of these dendritic cells to induce robust cytotoxic, T cell responses that are absolutely critical for killing cancer cells.
And are these different types of responses or just stronger responses? Like, what's the difference between maybe just upping the dose of the adjuvant that already exists for infectious disease versus a different adjuvant altogether?
Yeah, so you will actually require a combination of the activation of the both innate immune response as well as adaptive. And and in this particular case, you absolutely need the dual action to stimulate the innate immunity to kind of prime your immune system to drive strong enough of adaptive responses. Whereas in a lot of other vaccine types, they might not be absolutely required.
Okay, great.
Mm-hmm.
And for Omar, are there particular targets within the cancer cells or payloads that you're hoping to deliver with this approach that you're developing?
Yeah, I mean, we've been going through RNA sequencing traces for sort of the first cancer we've been looking at, so liver cancer with our collaborators. So we have different sort of patient-derived cell lines where we can look for commonly expressed RNA markers that we can target, and then we screen those to find ones that actually are expressed strong enough to activate the radar system. We are interested in the longer run also doing this kind of analysis across larger panels. Like, if we could find commonalities between like melanoma, lung cancer, liver cancer, right? Just to throw one out of the hat, like CMYC, for example, right? That would be interesting, I think, as well, because then you could just reuse the same machinery for multiple different types of tumors. But right now, we're very focused on specific targets in liver cancer, and primarily things that are overexpressed in liver cancer versus like normal like hepatocytes and other liver cells, because most of the delivery ends up in the liver anyway, so we can sort of constrain our target problem to that tissue. Yeah.
That was going to be one of my questions too. Yeah. Sorry, go ahead, Caleb.
No, are you able to do like non-coding RNAs too? Is that something you're thinking about? Because I guess you see all these like circular RNAs and all these kind of weird things showing up in tumors. I don't know if that's something you've thought about.
Yeah, we've targeted like link RNAs before. So yeah, not generally non-coding RNAs should be good. That's interesting actually. Yeah, we actually didn't look at non-coding RNA targets for the cancer work we're doing, but that would be interesting. Yeah.
Great. And are there any other cancers beyond? It sounds like liver is a good target because that's where it goes to anywhere. Are there other targets for cancer that this approach is more likely to work with than others?
Yeah, I mean, we've been mostly constrained by the delivery problem because we're focused very much on like in vitro transcribed mRNA, so like mRNA therapies, and so. I think we'd be interested in going after lung cancer or melanoma, and there are definitely groups that we collaborate with and talk to that are working on novel LNP formulations that can get to those cell types, although they're pretty new still and obviously not clinically validated. But I think that's the direction we would go in, is we would start to target those tissues next using novel delivery. Yeah.
Great. Yeah. Great. Well, we've got some audience questions coming in, so I'll move to some of those. We've got a question for Dr. Lareau. Can you conclude that those virus snippets detected in human blood are really a sign of functional viruses? And given that the piRNA pathway requires a repository of ancient viral remnants encoded in our DNA, could it be that what one detects just misexpressed snippets?
Yeah, it's a totally fair question, and I think this is also challenged the way that I think about this, because ostensibly these are discarded junk reads in your sequencing library, and so there's the sort of temptation to then think these are, again, kind of just like junk species because that's how we've treated them to date. What I can say is that there's definitely all kinds of confounding issues. For EBV, for example, the person that was pipetting to do the sample prep undoubtedly is EBV positive. They probably spilled some EBV just by breathing on the cell prep that they were doing for libraries. So really, I think this is underscoring the power of the sample size, where we're doing this for 500,000 plus people. And so the idea is that if there is real biological signal there, this is going to start to win out whenever you have these massive sample sizes. But if you were to look at, say, 10 people, then you would have all these confounding issues. So that's, I guess, like my first level. The fact that we see so much signal in these GWASs is reflective of the sample size driving that power. But whether these viral species would actually be kind of functional in a reactivated sense, all I can say is that this happens in humans, right? The fact that we have herpes virus infections early in life or norovirus infections early in life, then you go on and get a stem cell transplant. You're oftentimes viremic for these reactivation infections, which points to the fact that there are indeed, so maybe not in every case, but this certainly happens in humans where these are still functional species. So those are sort of the thoughts I have to that question, but it's a totally fair kind of button to push on, just because, again, this is all ostensibly junk, and like our ability to rescue it requires a stronger standard for actually showing function, and we're going to pursue exactly that.
Okay, so distinguishing the actual junk from what's having an impact is kind of the level of signal you see across different studies and samples and things like that.
Yeah, got it.
Great, and we've got a question for the Abudayyeh lab, Dr. Abudayyeh. Would there be an issue with titrating our would there be an issue with titrating out endo ADARs when using radar in vivo? That would free ADARs from editing the thousands to millions of ALUs, which might cause autoimmunity through non-editing dsRNAs. Any thoughts?
Yeah, I mean, it's always a concern, I guess, when you put, exogenous material into a cell. Like, how are you disrupting natural processes? I think, with this particular therapy, even if that was a concern, we're going after cancers where more often than not, right, ADARs are actually overexpressed. And even if we did disrupt endogenous ADAR, that might be beneficial, because it's, what you're disrupting is essentially the way for the cell to mask like double-stranded RNAs from like our innate immune system, which may be helpful in actually getting rid of the tumor cells more. But from just taking a step back, I mean, we haven't seen any serious issues with this. We haven't looked at it deeply, but I would imagine you would see some sort of innate immunity or toxicity if that were the case. And we looked at some of those pathways in the original paper. And another thing to think about is, the therapeutic RNA editing field has been working in this space for a long time, like oligos to recruit endogenous ADARs for therapeutic RNA editing. And that's a clinically validated modality now, right? Like, we have human data, we can get very efficient editing in the liver, and it doesn't seem like there's tox, and it actually works pretty well. So, and I would think that oligos are actually much more abundant than the mRNAs we're getting in the cell. So I would think, like, if there was an issue, you would see it there probably. But yeah, it's good to think about.
Yeah, it's nice you've got some preliminary research that it deals with some of those issues, so you don't have to be too afraid of those unintended consequences.
Yeah.
Great. And for Yzhong, you talked about having to make these cancer vaccine adjuvants much stronger. Obviously, if they're too strong, you're also going to run into some issues. So how do you strike that right balance? And can you do that prior to testing in animals? Is there a way to tell with the structures or any of what you're doing in terms of how do you get that balance before you even put it into animal models?
Yeah, exactly. That's exactly what we're trying to understand here. Thank you for that question, Shannon. So yeah, like you said, if the adjuvant is too strong, of course, can cause a lot of problems. Including maybe excessive inflammation, for example, systemic toxicity, and sometimes even immune suppression, which is not great. So it's absolutely important to strike the right balance between, potency and tolerability. And it remains one of the core challenges in adjuvant development. And like you also mentioned that, it's particularly important, especially in the development of cancer vaccines, because the patients sometimes have already been immunocompromised or, can have complex immune profiles at this point. And in fact, the potency of QS21, the molecule that we're studying, actually contributes to the potency of it, also contributes to the dose-limiting toxicity that we're seeing here. Then oftentimes you need formulation to actually reduce and neutralize such toxicity. So we're trying to understand, like I said, these molecules are highly glycosylated. We know these sugars are very important in how they interact with immune cells, how they can, bias the immune responses, and so on. So we're trying to develop, a biosynthetic platform to be able to just in a robust way to modify these structures to really understand how these sugars actually contribute to the activity or toxicity that we're seeing here, and then hopefully in the future to be able to make informed decisions before we put them into animal models, for example, and then to be able to develop these adjuvants that are both potent and safe to use.
Okay, so you can kind of use the glycans to tune the response a bit. Are there hints to which ones might dampen response versus make it stronger, and kind of some leads you're going to follow there?
Yeah, 100%. There are a couple of sites of the sugars that have been shown in literature that have significant outcomes and effects on the immune responses and the toxicity. Oftentimes, it just takes one sugar to change their profile. Actually, it's absolutely incredible. But traditionally, we very much lack a robust synthetic scheme to be able to modify these sugars, because if you think about those structures, right, if you remember your biochemistry chapter of carbohydrates, sugars look the same. So to be able to make glycosidic bonds selectively, you have to be able to selectively protect a couple of these hydroxy groups that are absolutely identical. Yeah, we are trying to be able to modify not only natural sugars, but also new to nature synthetic sugars to see how we can actually tune that and understand how they play in the immune responses.
Oh, interesting. I'll remind our audience, please continue submitting your questions and voting on others' questions you'd like to hear. We've got about 20 minutes left in the Q&A, so plenty of time to answer your questions. I have another question for Caleb. So you told us a bit about these anelloviruses. Are there members of that class we would have heard of in general or know about? What are the initial symptoms of those viruses? And why, is there any hints as to why they might cause autoimmune disease in more long-term situations?
Yeah, so the way that I think about these is if you just go through history in terms of how we've discovered viruses, and it's always because there's like an acute kind of phenotype, whether someone's coughing, they have a rash. In the case of, say, EBV, we actually discovered cells in a tumor where EBV has oncogenic transformative potential in creating a Burkitt's lymphoma. All of these are like, you see these kind of acute phenotypes, and you sort of have this immediate kind of Koch's postulates, cause and effect. This causes disease. I think more recently, if you think about the paradigm that's evolving with EBV and, say, multiple sclerosis, where ostensibly this virus is clearly doing something to the etiology of this disease, but the symptoms are not manifesting for decades. It sort of is challenging the way that you go to think about how do you find these viral species and what happens there. So with anellos, there's never been a kind of like rash or another kind of thing that would sort of more typically, lead us to think, oh yeah, this person's been infected and is sick in some way. Where these have been discovered are in the context of intense immunosuppression, where all of us have some degree of Anellos in our blood already, and then whatever is keeping them at bay seemingly is no longer intact whenever you're under, say, an immunodepleting chemotherapy. Now you start to see the viruses, but even then they were really discovered by not so much like isolating the virin, but like through PCR and sequencing, these kind of lower novel molecular tools. So the species, to your point about like maybe people have heard of, the predominant one in our blood are these things called torctinoviruses, but even then those aren't things that are covered in the New York Times. And so yeah, like I think it's a paradigm that through molecular tools that we're able to discover viruses that don't have this proximal phenotype is I think where this revolution is going to come. And I think Anellos are going to be one of the poster children of this, or at least that's where we're placing our bets that they're going to be. And then in terms of autoimmunity and whatnot, like it's important to emphasize that what I showed so far, what the field I think knows is it's purely a association, the sort of directionality with whether being high for Anellos is causal or maybe a consequence of having these is yet completely undetermined right now. But you can imagine that these viruses give you a whole source of foreign antigens that can be processed and presented on HLA. Thinking about the different risk alleles that are sort of universally associated with autoimmunity, being what our HLA genotypes are. You can envision some of these alleles being presented and processed differentially across humans. Maybe these then trigger autoreactive B and T cells, or not autoreactive, I should say, but anello-reactive. So that's kind of like my working model. But again, like before I started thinking about these, like probably in the last 18 months, like I had never heard of them. So I really think, this is why I'm going on seminars and symposia like this and screaming about Anellos, because I think they're grossly understudied, but I think they really do have potential to be kind of the next thing that we start studying more intensely for these kind of chronic inflammatory diseases that we don't fully understand.
Great, a lot to learn there. Yeah, look forward to seeing what comes out of it. And we've got an audience question for Omar. What would the effect of single A to I editing events in long non-coding RNAs to interfere with or enhance their function? Is that not the wrong direction?
Simple AI. It's an interesting question. I mean, I guess you mean from the perspective of off-target long non-coding RNA, because essentially with this technology, right, we're using the endogenous RNAs as like markers to trigger RNA editing of like a reporter we're putting in. So we're actually not editing necessarily like the non-coding RNAs or whatever we're sensing. And I guess if you have off-target AI editing, it kind of depends like what you're talking about. Like, yeah, there are AI editing sites that could disrupt like folding or enhance, maybe the activity of an endogenous interaction. It kind of depends on the site and where you're targeting. But apologies if I misunderstood the question.
Yeah, I think I thought about that a bit too, in that you're not changing the RNA that's already there. You're adding an RNA, and the RNA that's already there is activating the RNA that you've added.
Yeah.
It's around and around there. But I had a question about that as well, in terms of, so it's changing the RNA you've added and activating it. Is there a way to turn it off again? Or is that something you don't need to do or you're not concerned about?
Yeah, it's an interesting question. We've thought about the reversibility of it. It's hard to do that without getting too complex. You could imagine trying to think of ways to reverse the edit. But to be honest, because the RNA turns over so quickly, it's maybe lasting for 24 to 48 hours. The easiest way to have reversibility is just to let the RNA turn over. Yeah. And then with that said, I mean, in terms of controllability, generally we're not delivering the ADAR. You can also overexpress the ADAR and co-deliver it. And then there are ways you can control the ADAR that way as well. But typically we're trying to use endogenous ADAR. So, yeah.
Caleb asked a question of Omar earlier. Did anyone else have any questions for each other after hearing each other's talks that you're curious about?
I guess, Omar,
I was actually, well, I was just, I know nanoviruses have gotten some interest recently as like a gene delivery vector, and I was curious, because they were kind of sold as being kind of not having any biological effect on people. They're kind of just passenger viruses. But I guess your work is showing they could be, having effects on the body. I'd be curious your thoughts on that.
Yeah, I mean, I think as a whole, these idea of commensal viruses would, in principle, make far better gene therapy vectors. And again, I sort of the same belabored explanation I gave to the last question, where if you think about all the viral species we've discovered in the 1900s, things like AAVs, it's because we observed an acute phenotype. So it seems much stronger to use something that had no immediate acute phenotype to then use that to then deliver gene therapy payloads. I think that line of thinking is correct, and I think there are many other commensal viruses aside from just Anellos that would be really exciting gene therapy vectors. As far as I know, there's not been any clinical development of an Anello gene therapy vector, but if there were to happen, I would definitely suspect that there would be some sort of immune response to it. And I think it's probably something to do with like the degree of the viral infection, where you're sort of eliciting different immune responses. And if you load up someone with a giant gene therapy package, like that could definitely trigger something that is ordinarily silent with a kind of a more medium moderate dose that we're getting kind of endogenously. So I really like the concept. Like we should absolutely be mining these data sets for commensal viruses and use our gene therapy vector or discover gene therapy vectors that way. Based on what I've seen, I don't think Anellos are the right platform for that, but we definitely just need to continue developing them to know that better.
Yeah, cool.
And yeah, I guess to return volley question, maybe I've missed this, but the sort of programming gating, I know a lot about. I guess you kind of showed this AND gate capacity, maybe thought about like not gating and some of the other kind of like if then, whether that's relevant or not, or so could you speak to that sort of like more complex logic flow that you've considered with radars?
We have thought about trying to do like all the potential logic gates. I don't think we ever ended up mapping them all out, but there are ways to try to think about like not gates. I think we thought about ways to control like splicing of like a reporter or something, and that can change the output based on an edit. There's also combining this with some of the I think protease based sensors that I think. Was it Michael Elowitz maybe was developing, and also seeing Zhaoqing Gao, who also developed a similar technology. They do a lot with protease sensors. And I think you can combine protease sensors with RNA sensors and do much more complex logic as well, I think. When I think about the therapeutic translation, I think the more you layer on here, the less tractable it becomes as like an actual therapeutic solution. I think even like this simple method is hard enough. There's, issues with the best we can get with endogenous data right now is like 5 to 10% maybe of like, if you had to consider the promoter or just like a full, non-modified mRNA. And so like there's efficacy issues. I think cancer is nice because you only need like a little bit of like a toxic payload or immunogenic payload to work. But if you want to use this for like gene therapy and other stuff, that's where I think a lot of the work needs to go. But yeah, the logic stuff is cool. Yeah.
Great. I have another kind of general question for all of you in that all of your work, Yzhong already mentioned the interdisciplinary nature of her work, but it seems all of your work does have to bridge different, very vastly different fields. In Caleb's case, virology and data science. Omar, you've got this genetic engineering and cancer treatment. Can you speak to, for our younger audience members who are kind of just starting out in their career and how to kind of really make an impact with their work, speak to collaborations that contribute to your work? How do you find the right collaborators? How do you kind of develop those collaborations? Just kind of techniques like that, which play more into the full kind of trajectory of probably the rest of your careers as well, but what you're able to accomplish currently in these very innovative projects too. Yzhong, maybe you can start since you kind of touched on that already.
Yeah, of course. So I'm actually trained as a synthetic chemist in my PhD training. And then I kind of pivoted to synthetic biology for my postdoctoral training, and now kind of venturing into the field of immunology with the, just the combination of, academic training in molecules. In a way. So what that encourages me to do is to actually think about more complex biological problems on a molecular level. Because in chemistry, you set up a reaction, two things go in, one thing come out in an ideal case scenario. And then these are the type of mindset that I always carry with me to look at even more complex systems to see how we can actually understand all of these things at a molecular level. So that I think is very helpful, and in combination with the new unique tool set that I already have, to be able to combine these synthetic chemistry, synthetic biology, and immunotherapy, I guess at this point, pretending to be a expert in immunology here. But yeah, so I would highly encourage, trainees to expand their tool set, to be able to, try different things, different fields, and to think about problems that may seem very complex from a very interdisciplinary solution.
Great. Omar?
Yeah, no, I mean, I think, sort of a similar story. I was trained as kind of a bioengineer, and spent some time in medical school, although I did not finish. And so I kind of approach things from kind of like the medical need and, what's the therapeutic sort of unmet needs in specific spaces and work back from there. And while I think much more from an engineering and translation perspective, I think it's really helpful to have, other people who are, more familiar with diseases we're working on or the applications or delivery. And so a lot of our projects end up being sort of larger collaborations, right? Even for this particular project, we've been working with Dan Anderson and some other folks who work a lot with, LNP formulation for cancer delivery. We were working with Dan Duda, also at Mass General Brigham, who brings a lot of liver cancer and model expertise. And so it's a lot of fun because we get to, pull in people from different backgrounds and learn from them. And one thing I'll say is I think it's really helpful to put yourself in dynamic environments. Like I find Boston really exciting because it's we can go over to MIT for engineering expertise, but also the hospitals, and people are very willing to work on new problems if you just reach out to them and ask to meet. And so we've had a lot of success with that. But yeah.
Great. And Caleb?
I echo everything that's been said. I think just as a new investigator, it's becoming increasingly clear to me that the model of a single individual leading a lab where you're kind of in a vacuum working on something is just completely not consistent now with, I think, the way that any of us are running our groups. And you can see that from papers or sort of what we're talking about with different collaborators. And so I think it's important to just embrace it and recognize that this is where science is going. There's an added degree of complexity where you really do need to pull in multiple groups with expertise to do something transformative, and just not be afraid for that. Again, I also want to commend funding sources like this that sort of recognize that a lot of the traditional funding models that really are kind of the single investigator R01, it just doesn't quite fit with modern science as much. And so I think as much as we're able to have philanthropy fill this need in terms of how science is evolving, recognizing that people in the first stages of their career are having some really awesome ideas and just have difficulty getting some of the initial grants through these traditional mechanisms. Like this is where science is going. And so I guess both don't be afraid, and then also I encourage anyone that has the capacity to make more opportunities like this Michelson Award, and it really does make a huge difference.
Great. And yeah, given that you've all pretty recently just started your own labs, any advice for anyone who's about to do so or thinking about becoming an academic scientist? I know it's quite a change from being a trainee. So yeah, any thoughts about the challenges and advice for aspiring scientists? Yzhong, maybe you can start us off too.
Okay. Maybe my first advice would be don't sleep.
lots of work waiting to be but yeah, no. This of course, like you said, it's a very new experience, right? Instead of being a trainee, now you're a manager of the lab. You dictate where the science is going, and everybody in the lab as well. I think all the rumors that we have heard about getting tenure is extremely difficult. It's a lot of work. It's all true. But I also find it to be a highly rewarding experience to see how students start off, the lab starting from completely empty, and now it's fully occupied by, all the students and postdocs working very hard to generate results, to push projects forward. And students coming in with very little training background in synthetic biology, and now becoming experts and extremely familiar with the protocols. And just in a short, couple of months, really, it's a highly rewarding experience for me as a mentor.
Great. And Omar?
Yeah, I mean, I would say apply for a lot of grants. That was one thing, Fung gave us a lot of advice on when starting, and just like keep writing. And, I think these days we're doing like 80 to 90 a year. I think AI definitely makes it a lot easier to write grants now. So definitely, use the tools where you can. But I think we get rejected by a lot of stuff. And so I think the key is not to let the rejection get to you, and just, try to play the numbers here. And that's been fairly successful for us. I think also, trying to weather the storm. I think, one question we're getting a lot now is like, should we even start a lab today with everything that's going on? And, I think, there's still a lot of funding out there, beyond NIH. I think our lab is a third NIH right now. We have 3 R01s, but still, we get a lot of funding from, private foundations and other sources of funding out there. So I think also, trying to look really hard about all the opportunities out there and get on all the listservs is helpful. And then, yeah, I guess, that would be, I think, my main advice. I think that's what I spend most of my time on right now is trying to get funding for the group. So, yeah.
Sounds good. And Caleb?
My advice is that it's absolutely worth it. It's very challenging to do the job search. It's challenging to prove yourself as a grad student, and then again as a postdoc, and then still come up with a yet independent set of ideas to start the group. But the reward at the end, I mean, some of the mentoring young people, that's awesome, as my colleagues have indicated, is super rewarding too. But what I can say is that unlike basically any other job, you really do get to follow your scientific curiosity. For me, case in point with these Anellos, not something I wrote in my job description, my sort of proposed research statements. But what happens is you find something exciting that you get conviction about, and you have a ton of resources, you have autonomy, you have the ability to mobilize something that in basically any other career path, whether you're a industry science or even at your own startup, it's very difficult to sort of quickly iterate and quickly pivot. And so my advice is just don't lose courage. As Omar mentioned, it's a challenging time, but really is worth it if you're able to come to the other side. And certainly there's no shortage of challenges, but I think the scientific reward for being able to just follow your curiosity has been just so worth it for me. And I can't imagine doing any other job at this point.
All right. Well, very encouraging. Thank you all so much. That's all we have time for the discussion today. We will have a networking lounge immediately following this. So if you have further questions for our panelists or about the prize itself, please do join. Thank you all for joining us to discuss the future of vaccine science and immunology. This ePanel will be available on demand, and we will email you when the recording is available. The 2025 Michelson Prizes Next Generation Grant Application Portal is now open for those aspiring immunologists and vaccinologists out there to apply for next year's funding. And the portal closes on June 22nd, so get your applications in soon. You can find out more information through the link in the Event Information tab. In addition, we invite you to join us for our upcoming Vaccinology Conference, which is now available to view on live stream from the convenience of your own home. Tune in on June 5th for an exciting keynote address by Dr. Peter Hotez on vaccines and immunizations in a time of global boiling, megacities, and anti-science. There will also be research presentations by field leaders at CEPI, NIAID, and academic and industry scientists in vaccine design and innovation. Find out more on our homepage or in the link in the chat. And now we invite everyone to join us in the networking lounge to ask the speakers any remaining questions you might have and connect with fellow audience members to continue these conversations and strike up collaborations. In addition, we will have a representative from Michelson Prizes there to answer any questions you may have about the next generation grants themselves. The Remo platform allows you to jump between small group discussions as you would during a coffee break or cocktail hour. Click the button to join on the homepage or on the closing screen once we conclude. Thanks again for attending today, and we hope to see you all in the networking lounge.